Hi again, Biochem. So in the last video, we talked about the anaerobic pathway for cellular respiration. So if no oxygen is present, organisms can still break down glucose to make ATP. And something that every anaerobic pathway has in common is this process of glycolysis. So we consider glycolysis an anaerobic process where we break a glucose in half into pyruvic acid in order to make 2 ATP from that glucose. However, if you are an organism like a plant or an animal that has evolved aerobic respiration and has mitochondria to do it, and you're in a high oxygen environment, you are now able to go forward with aerobic respiration, which is able to produce much more ATP than anaerobic respiration. And that's what this video is going to focus on. So we have already talked about glycolysis. So we're going to assume glycolysis has already taken place. And now we are going to zoom in and look at the Krebs cycle. So just a quick orientation again about where are we in the cell. So imagine here we are in either a plant or an animal cell, right? We have already had our glucose gone through glycolysis to make 2 pyruvates out here in the cytoplasm. And now these 2 pyruvates are going to actually get transported into one of these mitochondria. Remember, both plant and animal cells have mitochondria and have to do cellular respiration. And so here we are. Here is the outer membrane of the mitochondria. And an important thing to note with mitochondria is just like chloroplasts, they're interesting organelles that actually have 2 layers of membrane. So there's this outer membrane that encloses it. That's what it's called, an outer membrane. And then in this lighter pink color, you'll notice there's also an inner membrane, and it's this super foldy inner membrane. So you can see how we have lots and lots of folds of inner membrane. That turns out to be very important for the electron transport chain. We're going to talk about that in just a minute. But for now, all I need you to notice is that this open gray space in the interior of the mitochondria is called the matrix, the mitochondrial matrix. Think about, I don't know, like the movie The Matrix. This is the space where it all happens. And so our pyruvates have been transported across 2 layers of membrane, both outer and inner, into the matrix. And this is where the Krebs cycle is going to take place. And just a quick reminder, if you're thinking about the chloroplast, sort of the equivalent place in the chloroplast would be the stroma. Just has a different weird name because we biologists love to create new vocab terms for you. Okay, so let's take a look at the Krebs cycle. So we just talked about this takes place in the mitochondrial matrix. What's going in is 2 pyruvates. And remember, those pyruvates are basically just like half a glucose. We've chopped a glucose in half. We've partially oxidized it just a little bit. And we have these 3 carbon pyruvates. We're also going to need some empty electron carriers to load up with electrons. So we're going to have some NAD plus and FAD that during the course of the Krebs cycle are going to get reduced. They're going to get electrons and hydrogens from those pyruvates to form NADH in the case of NAD plus and FADH2 in the case of FAD. And we're also going to make just a little bit of ATP. And we are going to take all of those carbons, they're going to be fully oxidized in this process and released as carbon dioxide, which is a waste product here. So let's take a look. So if you ever have to take like cell and molecular biology in college, you will have to learn an agonizing number of details in biochemical pathways. But for us, I really just want us to focus on the big picture. So here is the big picture. At the start of this process, and I'm going to go ahead and just put this all up here. So at the start of this process, we have 2 pyruvates. Each pyruvate is a 3-carbon molecule, right? We had 6 carbons in our glucose initially. Now we have 2 3-carbon pyruvates. And during the course of this complex series of reactions, the carbons in those pyruvates are going to be fully oxidized and released in the form of carbon dioxide. So at the end of this, we're going to release 6 CO2 molecules as a waste product, and that's where the carbon atoms are going. We're going to harvest a little bit of energy from the bonds in these pyruvates to make just 2 ATP. So not big beans here, but we have made a little ATP. However, the main purpose of the Krebs cycle is to harvest electrons. So we are going to be oxidizing, fully oxidizing all the carbons in these pyruvates in order to make activated electron carriers, right? So each NADH is going to have 2 electrons and a hydrogen transferred to it to form its reduced form. So NAD plus is going to be reduced to NADH. Here, and then also a little later in the process. And we also have the second electron carrier, remember from our first video, called FAD, that's going to be loaded up with 2 electrons and 2 hydrogens as well to make its reduced form FADH2. Right? So we have now, the big thing here is we have harvested, we've completely oxidized these carbons from pyruvate, harvested those electrons to make these activated electron carriers. Okay, so quick reminder, we've gone through glycolysis to snap our glucose in half and make a little bit of ATP. We've gone through the Krebs cycle in order to oxidize the carbons in pyruvate and transfer those electrons to these electron carriers, NADH and FADH2. And our next step is we're going to actually drop these electron carriers are going to drop off their electrons into the electron transport chain. All right, over here. And that is really what's going to power the vast majority of our ATP production. So let's take a look at that. So quick reminder, right? Krebs cycle, we have completely oxidized our glucose at this point. The carbons are released as CO2, and we have our electron carriers ready to go to the electron transport chain. Okay, take a moment on that table in the very last page of your notes to try to complete this second row for the Krebs cycle from memory. So pause the video and give that a try. Okay, let's check yourself. So hopefully you got this, right? Krebs cycle takes place in the mitochondrial matrix. The purpose is to basically break down pyruvate into CO2 in order to make our electron carriers, right? And a little bit of ATP. And here are our inputs and outputs. Okay, so remember how I talked about how these foldy parts of the inner membrane are really important? So this is where we're really going to get into that. So you will notice here that because of the folds in the mitochondria, in this inner membrane, we've created all these little enclosed spaces. And these little enclosed spaces are going to allow us to create a concentration gradient, right? So the whole goal of this process of the electron transport chain is to build up as large of a concentration gradient across the inner membrane as possible. So specifically, we're talking about a concentration gradient of protons, of H+. So we are going to try to get as much H+ into this little space in the intermembrane space. And by intermembrane space, I just mean the space between the two membranes, right? Between the outer and the inner membrane. So let's take a look at that. Just so you know, in the assignment that's posted, there is an animated Harvard video that I encourage you to watch. It goes into a lot more molecular detail than you need to know, but I think it really provides a useful visual. So if you're someone who finds you really need to see an animated visual to help you understand this, you might want to pause my video now and go and watch this video of the electron transport chain and then come back to this. But let's keep going here. So again, this is occurring across the inner membrane of the mitochondria. This is powered by the energy from the electrons carried by our activated electron carriers, so by our NADH and our FADH2. And that energy from the electrons is going to be used to pump H+ against their concentration gradient into that intermembrane space. And during this process, we're going to actually be reducing oxygen to make water. This is where the oxygen and the water in cellular respiration are going to come into play here. Okay, so big picture here, trying to move this out of the way slightly. The electron transport chain is using the energy of those electrons to pump H+ into this intermembrane space. Right? So again, if I'm orienting you here, this space down here in the bottom, this is the mitochondrial matrix. This is the interior of the mitochondria where we just did the Krebs cycle. And this space out here is the cytoplasm. So this is like the rest of the cell up here. Okay, so again, here we are. We're trying to pump protons out of the mitochondrial matrix and into this intermembrane space. So how are we going to do that? We're going to do that using the energy from these electrons that we harvested from the carbons and pyruvates in the Krebs cycle. So here we are. We're going to have NADH getting oxidized here, dropping off its electrons, and we're also going to have FADH2 getting oxidized and dropping off its electrons here. So both of these molecules in different complexes in the membrane are dropping off their electrons. And then those electrons are initially high energy electrons. They are going to gradually get drawn from complex to complex, and the energy of those high energy electrons is going to get used to power these proton pumps that are embedded in the inner membrane to be able to pump H plus against its concentration gradient and cause it to accumulate in this space between the two membranes. So if you actually look at this, and you don't need to know all these different protein complexes, it gets pretty complicated, but if you actually look at this, you're going to notice that each protein complex has a slightly higher electronegativity, essentially, which means it's going to attract those electrons more than the previous complex. And that's going to cause these electrons to get drawn from complex to complex to complex to complex. And each transfer is going to release a little energy to help power those proton pumps. At the very end of this, right, we have to think about, we have to draw, this is like a conveyor belt. There has to be somewhere for the electrons to go at the end. There has to be a most electronegative final location, because if this just stops, then our electron transport chain is done, right? No more H plus is going to be pumped. So we have to have a way to keep this moving. And the way we keep it moving is with oxygen. And that is why this is an aerobic process. This is where the aerobic piece comes in, the oxygen piece, because oxygen is actually one of the most electronegative elements in the universe, short of fluorine, right? And so oxygen can act as our most electronegative final electron acceptor. And so when those electrons get dumped onto the oxygen, it combines with two of those H pluses to form water, right? And so water is going to be our final location of the electrons, right? And it's just like a waste product that floats around. We're water-based organisms, no big deal there. So our oxygen is the final element that is reduced in this process. Quick reminder, remember that these electrons are getting drawn down the electron transport chain, and the whole purpose of that is to power the pumping of H+. And at this point, we have potential energy in the form of a gradient, and we already dealt with this with photosynthesis, right? Same exact thing with photosynthesis. We created potential energy in the form of a concentration gradient. And so think about this as like, over here in the intermembrane space, this is like a hydroelectric dam where we have a lot of water trapped behind the dam, and that water can be used as a form of energy to power a process that we want, namely making ATP. Okay, so I'd like you to take a moment to process that information. Oops, sorry about that. Take a moment to process that information with your notes. So fill out this next row in your table. All right, and let's check ourselves. So remember, the whole purpose of the electron transport chain is to harvest the energy in the electrons in order to pump H+ into the intermembrane space. And through this process, oxygen is going to be reduced. It's the final location of those electrons, and it will be combined with H+ to form water, which is just sort of a waste product of this process. This in of itself does not make any ATP, but it's creating the potential energy, right, from this H+ gradient to power the very last step of this process, ATP synthase, which makes an enormous amount of ATP. And we're going to talk about that in my last video.